HERVCOV, Ancient Viruses Among Us. Welcome to the HERVCOV podcast, voicing long COVID and its link to how contemporary viruses talk to ancient ones still roaming among us. In this second episode, we are going to meet endogenous retroviruses and discover their role in human evolution and health. Once upon a time, millions of years ago, there was a virus. This virus infected our ancestors, who then became humans, and over the course of evolution, it became inherited into our genetic information, also known as genome. These ancient viral relics, called human endogenous retroviruses, HERVs, stayed hidden for a long time until, during the COVID-19 pandemic, they were unveiled. A group of researchers from 5 European countries revealed the link between HERVs and severe SARS-CoV-2 infections, as well as post-COVID syndrome, including long COVID. Human endogenous retroviruses, or HERVs, are ancient viral sequences embedded in human DNA millions of years ago, through infections of our ancestors. Today, they make up around 8% of the human genome. For decades, scientists believed they were inactive genetic fossil. However, recent research suggests that some HERVs can be reactivated under specific conditions, including viral infections, inflammations, and immune dysregulation. In this episode, we explore what endogenous retroviruses are, how they become part of us, and why researchers are now studying their possible role in disease affecting the immune system and the brain. We will also discuss how HERVs may contribute to neurodevelopmental and neuropsychiatric disorders, including autism spectrum disorders, multiple sclerosis, and schizophrenia, as well as their possible connection with long COVID. What are HERVs and why are they important for understanding human health today?
As we mentioned in the introduction, human endogenous retroviruses of HERVs are ancient viral sequences that became integrated into the human genome millions of years ago. Although most HERVs have been inactivated over time by the accumulation of mutation, some retain the ability to be transcriptionally reactivated under specific conditions such as viral infection and inflammation. This has made HERVs an important area of research. Moreover, growing evidence suggests HERVs may act as a link between environmental triggers and the immune system and, when reactivated, can amplify inflammatory response and potentially contribute to long-lasting biological effects. This is particularly relevant for understanding complex disorders associated with chronic inflammation, including neurological, neuropsychiatric, and autoimmune disease, as well as long COVID.
How did the COVID-19 pandemic change research on endogenous retroviruses?
The COVID-19 pandemic greatly increased the interest in the role of HERVs in viral infection. Before 2020, HERVs were mainly studied in autoimmune disease, cancer, and neurological disorders. During the pandemic, HERV expression was detected in leukocytes of COVID-19 patients and associated with disease severity and requirement for respiratory support. These findings suggest that HERVs may contribute to immune and inflammatory response triggered by viral infection and have led to growing interest in their potential role in long-term post-viral complications.
What did the HERVCOV project discover about the relationship between SARS-CoV-2 and HERV activation?
The HERVCOV project has provided important evidence that SARS-CoV-2 infection can alter the expression of several human endogenous retroviruses and inflammatory mediators. Importantly, the project demonstrated that specific HERV family and immune-related markers were expressed not only in blood samples but also in nasopharyngeal swabs routinely collect for COVID-19 diagnosis. Their expression profiles were associated with disease severity, suggesting their potential value as biomarker of disease progression. Overall, these findings support the existence of biological link between SARS-CoV-2 infection, HERV activation, and immune alteration observed in COVID-19.
Could HERV activation can explain some long COVID symptoms?
Long COVID, also known as post-COVID condition, refers to the continuation or development of new symptoms for weeks or months after the acute phase of SARS-CoV-2 infection and is often associated with persistent immune activation, chronic inflammation, and immune dysregulation. An important question is whether in some individuals SARS-CoV-2 leaves an altered immune environment that can reactivate endogenous retroviruses and contribute to persistent inflammatory process. Growing evidence suggests that HERVs activation may also contribute to some long COVID symptoms. Preliminary studies of HERVCOV consortium have detected the presence of HERV-related marker in some individuals, raising the possibility that HERVs may sustain inflammation and immune dysregulation and contribute to symptoms such as fatigue, cognitive impairment, and neurological complaints. Although further research is needed, HERVs are increasingly being investigated as a potential contributors to biological mechanisms underlying long COVID.
What are the most important unanswered questions in HERVs research today?
Despite the significant progress made in recent years, many questions remain unanswered. Researchers are still working to understand the mechanism that trigger HERV activation and why these occur in some individuals but not in others. Another important challenge is to identify which HERV family are most relevant in different pathological conditions and how they interact with the immune system and inflammatory pathways. Finally, further studies are needed to determine whether HERVs can be reliably used as biomarkers of disease severity and progression, and whether they could eventually become targets for new therapeutic strategies.
Could understanding HERVs eventually lead to new therapies?
There is growing interest in the potential clinical application of HERVs research. One of the most promising areas in the identification of new therapeutic targets based on HERV-derived proteins and biological pathways they could influence. For example, several experimental therapeutic approaches targeting HERVs, including antibody-based strategies and antiretroviral therapies, have been investigated for their potential to reduce HERV activity and modulate its downstream effects. In parallel, measuring HERV expression may help identify patients at risk of severe disease or long-term complications, supporting a more personalized approach to patient care. Although further research is needed, these findings highlight the potential of HERVs as both biomarker and therapeutic targets.
In this episode, we explore endogenous retroviruses, ancient viral sequences that became part of human DNA millions of years ago. HERVs are now emerging as important players in immune regulation, brain development, and pathogenesis of different diseases. Researchers are investigating how their activation may contribute to neurodevelopmental disorders and how viral infections, including SARS-CoV-2, may reactivate these ancient elements. The study of HERVs is opening new perspectives in the understanding of the relationship between viruses, evolution, and human health. In the next episode, we will focus on long COVID and explore how persistent symptoms after SARS-CoV-2 infections may be connected to inflammations, immune dysregulation, and the activation of the endogenous retroviruses. Thank you for listening to this second episode about HERVCOV project. Stay tuned for more episodes where we shall proceed deeper into specific aspects of the project, including detailed insights into the research findings and their implications. The HERVCOV project has received funding from the European Union's Horizon Europe Research and Innovations Programme, with grant agreement number 10105732. HERVCOV brings together a consortium of esteemed partners. The partners include National Institute of Health and Medical Research, INSERM Transfer and Geneuro Innovation from France, National and Capodistrian University of Athens from Greece, Rome Tor Vergata University and Frascato Scienza from Italy, Aragonese Institute of Health Sciences and Aragonese Agency for Research and Development from Spain, Clinic and Hospital Center Rijeka from Croatia.